So either you people are not going back and revising whatever we do in the classes, or you people, all of you understand everything, which is the ideal situation, but I'm always doubtful of that situation. But if that is the case, that's the best I can ask for. Okay, so no doubts. Alia, Aisha, Mariam, all clear in chapter 6 so far?
Yes.
Yes. Okay. So who's going to give a quick recap of the last class? I think in the last class we talked about, let's start with transcription only. Application we did. Quickly, what do you understand by transcription? Let's start with Mariam. Mariam, would you like to talk about transcription? What is it? What do you understand by that? Are you there, Mariam? Am I audible to all of you?
Yes, sir.
Please, okay, please keep discussing. See, because otherwise I'll not know if I'm not audible or you're not speaking. Okay. Anyone else? Aisha or Alia, would you like to quickly, you can take the help of things on the board and can just give a quick recap of what is transcription. Whatever you understand, no pressures, okay? Just start it and I will keep pitching in wherever you are missing on to something or anything. Don't worry.
So
Yes, Alia. Yeah, go ahead.
So last class, we did transcription, which is the process of copying genetic information from only one strand of DNA into RNA. So
Perfect.
the enzyme we use here is a DNA-dependent RNA polymerase, which works from 5' to 3' direction.
Right.
So only one strand of DNA is copied because if both strands are copied, then it will code for two different types of mRNA, and they will have different sequences, and hence will produce two different types of proteins. So that, and then the second problem is that both RNA strands will be complementary to each other. So they will form double strand RNA, which will inhibit the process of the transcription and hence will be futile. So then we did transcription unit. It has three parts: a promoter, a structure gene, and a terminator. So the promoter is at 5 prime, 5' prime. If we talks about the coding strand, like there's coding strand and there's a template strand.
Template strand, right. So we you brought up a good point, Alia. So when we say a promoter or a terminator region, so we always take into account the template strand. Okay, so you will say that the promoter is towards the 3' of the template strand because that is where RNA pol is going to sit, right? So if you take the notation with respect to the coding strand, coding strand doesn't code for anything. We just call it coding strand because the mRNA that is coded will be having the same sequence as of the coding strand. But RNA pol, remember, works in a 5' to 3' direction. So obviously it will come and bind to the template strand, which is 3', which is running in 3' to 5' direction. So you can say that whatever is towards the 3' of a gene is called upstream. Okay. And whatever is towards the 5' is called downstream, like a river. So it starts at 3' where promoter is, and then transcription start site is also there. And then transcription happens. Polymerase slides all the way from 3' towards terminator region towards the 5'. So we call things towards 3' as upstream and sequences towards 5' as downstream. Okay. Is that clear?
Yes, sir. So,
Yep. Go ahead. Go ahead.
start.
Yeah, go ahead. Go ahead.
Okay. So there's a promoter. Here RNA polymerase is present. It will initiate the transcription. Like transcription will happen from here. Then there's a terminator, and the middle part is our structure gene. So we have, I think we have two types of structure gene. Like one is cistron, and there are types of cistron where there's a monocistron and there's a polycistron. So
No, but yeah, yes, right, Alia. You're right. But what is a cistron? Why have we
cistron,
brought in the word cistron? Or
yeah.
anyone
So
else who would like to contribute can just pitch in. Yes, Alia.
cistron is a segment of DNA that codes for polypeptide protein. So
So basically, it's a gene only, right? Right. So why don't we call it gene? Just we call it gene. We were calling things as gene, whatever codes for mRNA, we were calling it a gene very conveniently. So what happened that we had to bring up a new term? So that this is what you have to understand. See, if you just go for remembering stuff, there are too many things in biology to remember. For that matter, there are too many things in any subject to just remember. But remembering is not our goal here. You have to understand why we brought up the concept of a cistron. And on the basis of this concept, now we can divide structural genes into two different kind of arrangements. So, but before that, why we brought up cistron? Yes, Alia, you were saying something.
because I think it's a part of a structure gene, right? So like
You're right. So structural gene itself is cistron. We can call it, as you were going to say, we can call it polycistronic arrangement or monocistronic arrangement, correct? That you understand. That's there in the book. But remember, my question is, what is special about cistron? Why can't we just call it gene? Why we have to use another word, cistron? And we also talked about RNAs, type of RNAs. So what is transcription? If we go back to the definition, Alia, tell me what is transcription?
it is the process in which we copy the genetic information from one strand of DNA into RNA.
Into an RNA. Perfect. So basically, we are making RNA from DNA, right? So that is transcription. But later on, it was realized that there are more than one kinds of RNA that can be produced through transcription. There can be a mRNA, there can be a tRNA, there can be a rRNA. There are other forms of RNAs as well, which is beyond the scope of your syllabus at the moment. But so there's something called srRNA, snRNA. snRNA are small nuclear RNAs. So these RNAs stay in the nucleus and function there. They can bind to different segments of DNA, etc. srRNA is another form of RNA. LNC, non-long coding RNAs are there. But let's focus on these three. So we realize that there can be many different types of RNAs formed through transcription. But not all the RNAs give rise to protein, right? And remember the central dogma from DNA to RNA, RNA to protein. ultimately, if we are saying that this gene is showing a phenotype, is showing a phenotypic expression, that gene must be producing a protein. But we realize that all genes do not produce proteins. There are some genes that code for RNA, but that RNA does not go forward to make protein. For example, tRNA and rRNA. Does that make sense? Do you understand that?
Yes.
Yeah. That's why the concept of cistron came up. And cistron is a segment of the DNA that codes for a protein. So that DNA will get converted into RNA, and that RNA for sure will go on and produce a protein. Okay. And that RNA is called mRNA, the messenger RNA. Okay. So cistrons code for mRNAs only. Does that make sense, everyone? Yes, go ahead, Madhya.
So we have two types of cistron. One is monocistron, which is single cistron. It it's present in most of the eukaryotes. It has the interrupted coding sequence, like there will be exon, intron, exon, intron, and
Right.
so after the post transcription, these introns will be removed, and there will be only exons. So only exons will be present, and then again the transcription will be happened, and mRNA will be formed. So, uh, so the sequence of only extrons is present is in mature RNA only,
Yep.
and
Right.
it doesn't present in immature. Yeah. Okay.
So we call this kind of arrangement as a split gene arrangement. So eukaryotes do not have their genes coded in one continuous manner. Okay. Every gene of ours have intron and exon. Now, why is that and what advantage it gives? There are many hypotheses and theories around it. And some of these theories are being tested or being looked forward, which of the theories make more sense. But the fact that you have an intron and an exon, and then you have to cut out all the introns during post-transcriptional modifications after transcription is a very energy consuming process. We are using lots and lots of energy just to make mRNA, right? Whereas prokaryotes, bacteria, are very simple. They have a polycistronic gene arrangement, structural gene arrangement. And what is polycistronic? Iram has joined us. Iram, would you like to help Alia here and talk about polycistronic arrangement? Are you there, Iram? How are you doing?
Yes, sir. Yes, a polycystronic structural gene is present in prokaryotes. Here, the coded info is present in a continuous manner.
Right. So so it's present in a continuous manner, but that's it. But why does it called polycistronic? Anything special about this arrangement? People, we discussed all of this, but you people are not revising, it seems.
Because many genes will be formed, like many
Uh-huh.
RNAs will be formed from it. Like in monocystonic, only one mRNA is formed, whereas in poly there will be many mRNAs. Like shown in diagram, there are 3 mRNAs will be formed.
Right. So mono means single, poly means multiple, and cistronic means a region of DNA that give rise to a protein. So basically, a polycistronic arrangement is like a cassette where multiple genes can be fit under a single promoter and a single terminator. So once transcription begins from the promoter, it will only end after translating all the genes which are there in that polycistronic arrangement. Okay, so we do not need promoter and terminator for each gene here, each mRNA here. They are together, but ultimately they will form 3 different mRNAs, right? It saves space, like we talked about in the last class. Whereas in eukaryotic system, like humans, for every gene which will translate into protein, there will be a promoter, a separate promoter of itself and a terminator region. And the gene itself will be in a split gene arrangement, having introns and exons, right? So this is the difference, the major difference between prokaryotes and eukaryotes. Is it clear, everyone?
Yes, sir.
Then we talked about this transcription in prokaryotes, which is pretty straightforward. There are 3 steps: initiation, elongation, and termination. Initiation is where the sigma factor guides the RNA polymerase to go and bind to the promoter region, and the transcription begins. And as it continues towards terminator, the RNA is formed. It keeps on elongating. So the second step is elongation. And once it reaches towards the terminator region, a different factor called rho comes and binds. And remember, sigma detached from the RNA pol the moment elongation started. So sigma is not important for elongation. It is just important for initiation. And for termination, another factor called rho, which is termination factor, comes and bind to RNA polymerase. And the RNA is released or detached from the polymerase, and the polymerase itself is detached from the DNA segment. Okay. So this is how one transcription unit completes. Now, in today's class, we will start with some complexities which are there in eukaryotes. So we'll start with transcription in eukaryotes. So in eukaryotes, let's start with this. So far, so good. Everything clear to everyone? Okay, transcription in eukaryotes. Now in eukaryotes, there are 2 complexities, okay, as compared to prokaryotes. So there are 2 complexities. First complexity is that there are multiple RNA polymerases present in eukaryotic system. Okay. So whereas in a prokaryotic system, there is only one RNA pol. Okay. So there are multiple RNA polymerases. To name specifically 3. Okay. Polymerase 1, 2, and 3. And you have to also know here that in eukaryote, they do different functions. So I'll write pol 1. So RNA pol 1, it helps in the transcription of rRNAs. So you can write it transcribes. Remember, I told you there are many different kinds of RNAs that are transcribed. So in prokaryote, the single polymerase transcribes all kinds of RNAs, but in eukaryotes, RNA pol 1 specifically transcribes rRNA. And what is rRNA? Ribosomal RNAs, right? And you have studied in your junior classes that ribosomes, what kind of a structure our ribosome has. Remember, it has a bigger subunit and a smaller subunit, right?
Yeah.
Yeah. So rRNAs also are of different types. So most common ones are 28S, 18S, and 6S, but more specifically, it's called 5.8S or 6S. Okay, so these are the three. S means the sedimentation coefficient, right? That you have studied already. So this is what RNA pol 1 does. Then there is a RNA pol 2. And this is a very important one because RNA pol 2 is responsible for making mRNA. Okay. But as I told you, in eukaryotes, you cannot directly make a mRNA, right? Whatever you make by transcribing the DNA, it contains both exons and introns, right? So when that is formed, we do not call it a mRNA because it's not ready with the message. The message will only be completed and clear when you will remove all the introns and stitch all the exons back together. Okay. For that, let me do this. Stopping the share for a moment. Can you all open your NCRT and come to this figure which shows transcription in eukaryotes quickly? Everyone? Let me pull it out. Yep. Did you open your books, notebooks, people? Yeah. Can you all see the screen that I am sharing?
Yes, sir.
Yeah, this is the figure I'm talking about. Can you all come to this figure? So look here. This is transcription in eukaryotes. And if you pay attention, so this is your eukaryotic DNA, right? Here also, there is an initiation that happened. So there are three stages here also: initiation, elongation, and termination. But the factors are different because not like it's not sigma and rho here. They're called, they have different names and different structures, but you will find similar kind of proteins present in eukaryotic system as well. But just remember that there is an initiation factor and there is a termination factor. But what you should pay attention to is this RNA that's coming out, right? So this RNA that's coming out, you can see that it has two different colored regions. One is this yellow, orange, yellow, orange, yellow, orange, right? It comes like this. And the yellow one is marked here as exons. And the orange ones are introns. And you know that ultimately these introns have to be removed. So how are they removed? They are cut at two edges and then converted into a round circular RNA. And then whatever is left is all the exons, and that is stitched together. All the exons are stitched together. So here you can see everything is yellow, no orange. So no intron is present. All introns have been removed. Okay. Now the question is, which is a very interesting question, when introns ultimately have to be removed only, why are they present in the first place? Why are they? It's like you have put a message, you have written a letter to your friend, but after every word that makes sense, you have put a word that does not make sense. And if your friend knows how to decode the letter, then only he can understand the letter, right? Do you understand what I'm saying? Am I audible, people?
Yes, sir.
Yes, sir.
Yeah. Okay. So this RNA, we do not call it mRNA when it comes out, okay? Though it's written here as mRNA, but it's the true name for this kind of RNA where there is exon, intron structure is called hnRNA. So this is called hnRNA. Okay. And what does hnRNA stands for? It's called heterogeneous nuclear RNA, because at this moment it is in the nucleus only. And it's called heterogeneous because there are two different kinds of sequences present. hnRNA, heterogeneous nuclear RNA. It has exons and it has introns. Removing of introns is called RNA splicing. It's a post-processing of the RNA after transcription is over. And with the help of splicing, you are actually splicing out all the introns. So they go away and they're not required. And then all the exons are together stitched. And this is a mature RNA. And we call it mRNA or messenger RNA. So only when it gets mature, it's called mRNA. And remember, what is this green thing? So you see two other words here. One is called capping and one is called tail. So capping and tailing. This RNA, when it comes out, we know that RNA is not very, very stable, a molecule. So, and also eukaryotic RNAs, which are heterogeneous nuclear RNAs, they're longer also. The genes are longer also because more than half of the gene is just intron sometimes, okay, which has to be removed. So it will take time, right, to be transcribed completely. So capping is and tailing is the process by which you make the RNA stable, because you still have to work on it, right, and make it a mature mRNA. And to work, it has to be stable. It should not just start degrading. So capping is put a cap at the 5' end. Remember, RNA polymerase works from 5' to 3' direction. So the first thing that comes out is a 5' end. So at the 5' end, capping takes place. Okay, capping is nothing but at the 5' end, you put this MG3P. So these are 3 phosphates, PPP, attached to a methylated guanine. So it's called MG3P, MGPPP. So that is the cap. You put a cap on it, and then you put a tail. Tail is nothing but long sequence of A, adenine, A. So lots of A are attached. When I say A, it's not the alphabet A, but adenine molecule is attached. So it's called polyadenylation. Okay, tailing process is called polyadenylation. These two things at the front and at the back of the mRNA keeps it stable and protected, prevents it from degradation. Is that clear, everyone?
Yes.
Yeah. So this splicing happens, mature mRNA comes, and this is done by this polymerase is RNA pol 2. So RNA pol 2 transcribes, let's go back and write, it transcribes mRNA. But before that, write hnRNA, because it transcribes hnRNA. This is immature, which later becomes processed, or we call it splicing, capping, tailing, and splicing. There are 3 things that happen in post-processing, post-transcriptional modification, we call it. So in post-transcriptional modification of the RNA, there are 3 things that happen, 3 processes. One is capping, then tailing, and then splicing. Okay, so processed or spliced is formed mRNA. So in the options, if suppose this question comes in entrance and they say that RNA pol 2 transcribes, and in the option there is no mRNA. So the option is rRNA, tRNA, hnRNA, none of the above. So what common mistake mostly students do is They think that Pol2 makes mRNA and mRNA is not in the option. So it could be the fourth one, which says none of the above, but that's not the right answer. The right answer is hnRNA. Okay, heterogeneous nuclear RNA, because that becomes mRNA after processing and post-transcriptional modification. Is that clear, everyone? Okay. Now, the third polymerase that eukaryotes have is RNA pol 3, RNA polymerase 3. And what does this do? It is responsible for transcribing tRNA. Okay. So it transcribes tRNA. Also, it transcribes srRNA and snRNA. These are small nuclear RNAs, but main is, remember, tRNA. So now you can see in eukaryotes, all the RNA which are responsible for the final step, which is translation, which is tRNA, mRNA, and rRNA, all these are transcribed through different polymerases. Whereas in prokaryotes, it's done by the same polymerase, so no problem. So this is first complexity, that it has multiple RNA pols. The second complexity is, which let me write here. Can anyone think what is the second complexity? We talked about it just now. The second complexity is that the primary transcript. When I say primary transcript, it means the first thing that comes out of the polymerase enzyme. This is the primary transcript, is a heterogeneous RNA. So the primary transcripts are product of nothing but RNAs. Okay, it's a name for RNA. Why they are called transcript? Because they are products of transcription. Is it clear, everyone? So basically a transcript and an RNA means same thing. Okay, cool. The second complexity is that primary transcripts contains both exons and introns. Okay. And because it contains exons and introns, this primary transcript is non-functional. You cannot use this as a RNA and give it to ribosome to make a protein. Okay, ribosomes won't be able to make a protein out of this primary transcript. They cannot work. This will not work unless you remove all the introns and stitch all the exons back together. Okay, so write down these two things. There are two complexities in a eukaryotic system. There are two complexities in the eukaryotic system. First is there are three RNA polymerases. There are three RNA polymerases which transcribes for different kinds of RNAs. Which transcribes for different kinds of RNAs. RNA pol 1, as it is written, it transcribes rRNA. Pol 2 transcribes mRNA through hnRNA, and pol 3 transcribes tRNA. So this is first thing. Second complexity is that the primary transcript, or the primary RNA that comes out of the nucleus, contains both exons and introns. Contains both exons and introns and is non-functional RNA. It is a non-functional RNA. So what is the solution for this? If you have guessed by now, then you are right. The solution is post transcriptional processing. Okay, and this includes, this includes three things. One is capping. You put a cap. Capping is 5. Sorry. Capping is towards the 5 prime. It happens at the 5 prime, and it is simply methylated guanine triphosphate, MEG3 repeats triphosphate. The second thing that happens is tailing. Put a protective tail, and this is simply polyadenylation. Put a long chain of AAAA, somewhere around 200 to 300 molecules are put at the tail to protect it. And the third thing is splicing. This is the most important thing. Splicing is where you cut out introns. So introns are out and exons are in or stitched together. In and out. Is it clear, everyone?
Yes, sir.
Yeah. Look at this figure, because this figure, you can be asked to make this diagram in exam. So look at it and let me know if any part of it is unclear to you. I give you 1 minute, and also to understand the two complexities. Is it clear to all of you?
Yes, sir.
Okay, now let me come back to this question so that you can build some good concepts. So in eukaryotes, why do you think eukaryotes have this add-on additional complexities? Why didn't we learn from bacteria? Be simple, put multiple genes under one promoter. The job is easy. Why? Why do we have to be so complex? What purpose does it serve? Anyone? Any guess? So now with recent studies and research work into the RNA biology, we started to understand that this complexity of having a split gene arrangement and the need for splicing is not just by chance present. Okay, it actually gives us a hint, or it is you can call it a reminiscent of the RNA world. Okay, so at one point of time, RNA and RNA dependent processes were the major processes in living system, and they were most important. Now, the benefit of this kind of arrangement is regulation of gene expression. Okay, suppose if I do the splicing in two different ways. Okay, because of splicing, when we are cutting out introns, let's say a very small amount of exon is also cut out in one case, whereas in the other case, it's not cut out. So don't you think that the two proteins now formed, sorry, the two mRNAs now formed will be slightly varied of each other? So they will be called splice variants. So the concept of splice variants have told us that the same protein can have two different splice variants that can function in slightly or very different manner. So both the proteins comes from the same gene, but depending on which splice variant is formed more in the cell, it can do a different function. It can regulate a different pathway. Does it make sense? That's why more complex organisms like eukaryotes have this complex arrangement where they can achieve this complex regulation. Okay. Whereas prokaryotes do not have this complexity. Okay. Does that make sense, everyone?
Yes.
Okay, perfect. So if this part is clear, then before we go to translation, I would like to talk about some things related to the genetic code itself. So what do you think is a genetic code in both the processes that we have studied till now, which is replication and transcription? What are we doing? What is the cell doing? Can you tell me quickly? What is the gist in shortcut, if you want to tell me? What is happening in both transcription and replication?
In replication, the DNA is replicated to DNA, and in transcription,
Right.
RNA is DNA is one strand of DNA is replicated to RNA.
Yeah. So this is the difference, right? You're saying this is the definition also, that replication is DNA to DNA and transcription is DNA to RNA. But I'm asking common thing. What is happening if you just break it down? For example, if I just ask you in nature, if we talk about the food system, the food web, what is happening? So what is happening is one living system is depending on another living system for its energy, right? Except for chemosynthetic bacteria or autotrophs, which can derive energy from chemicals and sunlight, everything else relies on other living systems. So whether it's a herbivore organism also eats another life, which is a plant life. A carnivore eats another life, which is an animal life. So if I ask you what's happening in general in nature, you can see that one kind of a living system is killing the other kind of a living system and eating it up so that it can produce energy inside its own body. Do you understand?
Yes.
Right? Yeah. So if I ask you what's happening basically in replication and transcription, what is the common theme there? The common theme of jungle is either predate or get predated. Yeah. Either you become the predator or the prey. And when I say predator and prey, I'm not only saying that animals are predators and plants are preys. You know, plants also which are predator plants. There are carnivorous plants as well, about which you have studied, like Venus flytrap, right? Pitcher plant. So these are carnivorous plants that kill animals and eat them. Nature is full of things. So what's the common theme of replication and transcription? Quickly. Alia? Close your eyes and think what's happening there.
One nucleic acid is converting to another.
There's no conversion actually, if you pay attention. Nothing is being converted to anything. The original DNA, the parent DNA, is not converting into the new DNA. What is happening? Conversion. Only one thing. What happens when you miss a day of school? The next day you go and you ask your friend for his or her notebook. You bring his or her notebook home. Do you convert his notebook into your notebook? You remove the sticker, put your sticker and write your name, and from next day it's your notebook?
We copy it.
Exactly. So the common theme here, whether it's replication or whether it's transcription, is copying, right? In replication, the two strands of DNA open, and in a semi-conservative method, the new strand is copied from the old strand. Do you understand that? That copying is on a complementary basis, in both the cases, right? So when you copy from your friend's notebook, you do not do complementary copying, you do exact copying. That's the difference. Here it's a complementary copying, right? But it is simply copying only. So the common theme of replication and transcription is that a nucleic acid was copied to form another nucleic acid. Right now, this is the common theme. So till now, it was very simple rule. If you see A, and if you're making a DNA, put a T. If you're making an RNA, put a U. If you see a G, put a C. If you see a C, put a G, and so on, right? Simple rule. So it was very easy to understand because complementarity, the rule of complementarity was helping us. Now, as we go from transcription to translation, the process of translation is not very straightforward. And scientists also invested a lot of time understanding how a protein is formed from an mRNA. And it looks like how many base pairs are there? How many nitrogenous bases are there? How many nitrogenous bases are there in nucleic acids? DNA, RNA combined. Yes, Alia, you were saying something?
5.
5, right? But at one time, there can be 4 only, whether it's a DNA or RNA, right? There is no hybrid of a DNA and RNA where T and U are present together. So you are right. In totality, there are 5 nitrogenous bases: thymine, uracil, ATGCU. But either ATGC will be present or AUGC will be present, right? So let's say 4 nitrogenous bases are present in RNA, right? How many amino acids are present in a protein? How many different kinds, I'm asking? How many types of amino acids are there? 20. Yes, that's correct. So types of nitrogenous bases in a RNA, which will be studied, is 4: A, U, G, and C. And types of amino acids in any protein, okay, is 20. To be exact, it is 22 now. We have discovered 2 more amino acids, but those are not very unique amino acids. Those are just modified forms of already existing amino acids from the 20. Okay, so the 2 extra amino acids are not something very unique and different. So that's why we do not study it at this level. But if anyone is interested, please go and find out what are the 2 extra amino acids. Anyone knows, by the way? So these are called rare amino acids, and we call them rare, and also it's named as the 21st and the 22nd amino acid. Anyone knows, by the way, the name? No? Okay. So there's amino acid called cysteine, and there is amino acid called lysine, which are part of these 20 amino acids. Then these 20 amino acids are also, subdivided further on the basis of essential amino acids and non-essential amino acids. So non-essential amino acids are those which, we easily produce. Essential amino acids are those which are required and the supply is not very abundant through diet or other sources. So cysteine and lysine are 2 amino acids as part of these 20. There are 2 modified versions found called selenocysteine, okay, and pyrrolysine. So selenocysteine and pyrrolysine are known as the 21st and the 22nd amino acid in the genetic code, but you don't have to read about the extra 2. So for you, there are 20 amino acids. Now pay attention here. 4 nitrogenous bases and 20 amino acids. Now that's a problem. If there were just 4 amino acids, again, the law of copying would have worked, right? Let's say there were just 4 amino acids. So A will code for one, U will code for the other, T will code for the other. And so when ribosome will read A, U, G, C, it will put one of the 4 amino acid in the chain. But that's not the case. There are 20 amino acids. Now that's a problem. Now, can you tell me logically, and it was also not a biologist, okay? So there were lots of people who tried to understand, how does it make sense? How does 4 nitrogenous bases can code for 20 different amino acids? How is this code working? What is the rule of this code? So if you just have, let me ask you, how many of you are interested in ciphers? Cipher language? Anyone knows what is ciphering? No? Or how many of you know about Morse code? Anyone knows about Morse code? Why it is called a code? Yes, Alia knows Morse code. Yes, Alia, what do you know about Morse code?
I've just heard about it.
What have we heard about it? Let's start there. I'm sure you all are very smart. I don't know why you don't speak. I had a class once that was full of people who wanted to speak and never stop. There, the difficulty was that I could not make them shut their mouths. Here, the difficulty is I cannot make you people speak up. Some of you do speak, so please, all of you speak, and that will be great. Yes, Alia, what do you know about Morse code and why it is called a code? See, there is an easy way to teach biology, the internet is full of it. You don't have to spend a penny, believe me, if you want to learn biology the boring way. You have NCRT, you have YouTubes, you have thousands of people out there teaching you things, but think to yourself when every day you turn towards Learnivio or any such platform where you come in contact with a live performing teacher sitting thousands of miles away from you, talking to you and trying to put something in your head. Why are you doing this? And on top of it, you're paying for it. Why? Because it's not for the knowledge. Knowledge is free. It's for the way this knowledge is going to stick in your head. You're paying to interact with the teacher. Otherwise, just put genetic code on internet. A Wikipedia page will come containing 20 pages. Read it all up and you will know what genetic code is. But that's not how education works, right? There always have been a concept of a teacher who's just simply a person who's a little bit senior in procuring knowledge, and then it passes it on to someone else who's called a junior student, right? And these roles are reversible. So I may be a teacher for you, but I'm a student for someone like my supervisors and professors. You are a student for me, but you can be a teacher for someone else who does not know what you know. It's simple. So if you are paying for that, why don't you participate in that? Yes, Alia, what do you know about Morse code?
So it's kind of a secret language type. Like it was
Mhm.
you. I've seen in a series. So they were talking in Morse code. Like there are dots and there are hyphens and the way
Yes.
we can.
So Morse, you're right. So what the most important point you brought up is Morse code works on dot and dash. Just two things. It's a dot dash language. It's the sequence. So how using just two things, a dot and a dash, you can create a language? How can you code all complex information in a dot or a dash? Another example I can give you is the binary language of computer, which is just 0 and 1. You know that everything that you do on a computer, the language of computer is just 0 and 1, right? Do you know that or not? Called binary language. Even sign languages, there are other different kinds of codes. So in Morse code or in binary language, the trick is how you arrange it. So just by producing dash and dot, it's like if it is like dot, 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 dash, dot, 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 dash, dash, dot, it means something. But if it is dot, 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 dash, dash, it means something else. And also the duration pause between one continuous signal and the other also tells you something unique about that language, right? So if you have to understand that code, you have to know the rules of that code. Similarly, if you have to understand how RNA codes for a protein, you will have to first understand how these four amino acid, how these four nitrogenous bases, just like the two symbols in a Morse code, can code for 20 different amino acids. So biologists tried, there were chemists, biochemists, geneticists, a lot of people came together, try to understand, try to decipher this genetic code. It was very, very challenging. Okay. Finally, it was a physicist. Okay. Physicist means person who's doing research in physics. His name was George Gamow. Okay. Wait. Yep. George Gamow. It's the same Gamow. Remember, I showed you a book written by Watson called Genes, Girls, and Gamow. Right. So George Gamow, he for the first time put an argument forward in the scientific community. He said that, see, you people are saying that you just have 4 bases. And then those 4 bases somehow code for 20 amino acids, right? Then I think mathematically it's only possible if the code is made up of 3 nucleotides. So one code is made up of 3 nucleotides. Okay, then it makes sense. And then it can code for all your 20 amino acids. Do you understand what he said? He said that ATG, no T is there in RNA, so my bad. ACG, if it is present in this sequence, if this is the sequence ACG, CAUUAACG, if this is the sequence, George Gamow said that first 3 is one code, then second 3 is another code, CAU, then UAA, and then CG, and let's say there is a A here, CGA, this is another code. So he said each code constitutes 3 nitrogenous bases. And people ask, okay, why 3? Why not 2 or 1? See, 1, it will not make sense, because if it is just 1, you just have 4 nitrogenous bases. Why not 2? Because
It will
mathematically,
have 16.
sorry, it will have 16.
16.
Oh, right. So how did you do that?
4 into 4.
Yes. So if the
4 to
code
the
is
power
1, yes. So there are 4 total nitrogenous bases. If you are saying that the code is made up of 1, then it will be 4 to the power 1, which is simply 4, right? So you can only code for 4 amino acids. We already know by doing protein analysis that proteins have 20 amino acids in them. Then someone said the same thing. If a code is made up of 2 amino acids, so 2 nitrogenous bases, it will still be 4 into 4, which is 16 amino acids. That also can't be the case because we already have 20 amino acids. Then that's why Gamow said, see, it has to be 3 nucleotides or 3 nitrogenous bases together coding for 1 amino acid. That way it will be 4 into 4 into 4, which is 64 amino acids. Now he said that this way, if you make a code of 2 nucleotides, you will end up short just 16 amino acids. But the problem is if you make a code of 3 nucleotides, you will have to make 64 amino acids. But do we have 64 amino acids? No, we only have 20. So back then, people thought maybe we have just discovered 20. We will find out more. And maybe the number will go up. Okay. Then someone said, what if it goes beyond 64 then? Then you will say the code is made up of 4. But then people realize that whatever protein they take from wherever in the eukaryotic system, whether it is animal protein, plant protein, from anywhere, they just constituted those 20 amino acids. Okay. And when I say now 22, those are again a modified version of the already existing amino acids. So let's say 2022. Till it is less than 64. Now the next question was, why 60? What happens to 64 codes then? So it will generate 64 codons. So we call each of it, let me erase this. And so ACG is called one codon. Then CAU will be another codon. This is codon 1, this is codon 2. Okay. So similarly, there will be 64 codons generated, but there won't be 64 amino acids. So we remove this term from here because we know that there is not 64 amino acids, and we use the word codons. So here we can just make 4 codons. Here we can just make 16 codons. Here we can just make, sorry, not just, here we can make 64 codons. Now, it is not very difficult to understand that 64 codons can be used, and each codon was a triplet. Okay. Then came a scientist who's our very own. His name is Hargobind Khorana. How many of you know Khorana? Professor Khorana, the eminent biochemist of India. So Hargobind Khorana was synthesizing RNA molecules with defined combination of bases. He said that if what Gamo said is true, I should be able to use codons and synthesize RNA molecules. Okay. In a cell-free system. So other people like Marshall Nirenberg and all, they also, synthesized proteins in a cell-free system. You don't need a cell to make protein. You can make protein because you know the code now. You can just make protein if you have the mRNA and ribosome. Okay. Now the question is, how does this 64 codons code for 20 amino acids? And the answer is very simple. Any kid can guess the answer. Tell me, how will you adjust 64 students into 20 groups? It's simple. Each group will have more than one student. Correct? Do you understand, everyone?
Yes, sir.
So what they figured out is, I think it's in your book, a figure called the codons for amino acid or something. Can you come to that page and also let me know what is that page number? Let me pull that figure out. Anyone can tell me the page number as well?
112.
112. Okay. Just wait. Okay. Let me pull it out for you. Yes, I found it. And it's here. Can you all see the screen that I am sharing? Yes, no?
Yes.
I think you, yeah. So this is the figure. Anyone who understood the figure just by looking at it? It's a very simple one. Yep. Anyone can explain this to me, what's on your board or what's in your textbooks? Come on. Anyone? I'm sure that you all understand what this figure is, right? Okay, let's see. So this figure or this table tells you about the codons that code for various amino acids. Now, on this axis, there is this first position. So Gamo said that a codon is a triplet of nucleotide or base pair, nitrogenous bases. So the first position in that triplet is here, UCAG, because these are the only 4 nucleotides possible in RNA. The second position is given on this axis, and then third position on this side. So if you see U, and here is the first U. So the first one is UUU. So uracil, uracil, uracil. If all three are uracil in a codon, the amino acid that will be formed is PHE called phenylalanine. Okay. So the 20 amino acids, PHE is phenylalanine, LEU is leucine. So you can see for phenylalanine, there are just two. So two different codons can code for phenylalanine. Either it's UUU or UUC. Okay, but if it is UUA or UUG, that will not code for phenylalanine, but it will code for leucine. LEU is leucine, along with 4 other. So for leucine, there are 6. So it's also again not same number. There are 2 codons that can code for phenylalanine, but there are 6 codons that can code for leucine. And look at what's different in all the 6 codons. Either it could be UUA or UUG. What is the difference in UUA and UUG? The third position, right? Yes or no?
Yes, sir.
Similarly, if you just look at this box, CUU, CUC, CUA, CUG, where is the difference?
The third position.
Again in the third position, right?
Yes, sir.
Look for the ILE is isoleucine. There are 3 codons for isoleucine. Again, AUU, AUC, AUA. Where is the difference? In the third position. So if you will see for all the 20 amino acids, phenylalanine, leucine, isoleucine, methionine, valine, serine, proline, threonine, alanine, tyrosine, histidine, glutamic acid, and aspartic acid, lysine, aspartate, all of these amino acids, the difference in their codons will be on the third position. Now, it also makes sense. Why? Because the third position of every codon is more prone to mutations. It has been seen that mostly chances of mutation are on third. And let's say, let's do this exercise. Let's say the original code was AGU. So what will it be coding for? Quickly. If it is AGU, what will it code for? Who will tell me first?
SER.
SER is serine. Okay. So it will code for serine. You also, those who did fast, they did it because they knew how to read the table. So AGU codes for the amino acid serine, right? Now let's say the U, the AGU, the U part got mutated to C. It became AGC. Now, will there be a problem in the protein? If AGU becomes AGC, what will happen? Come on, look at it. AGU mutated to AGC. What happens?
It remains same.
It remains same. It remains serine only. But there is a mutation, but there is no change in the protein. So protein got saved, right? What will happen if AUU is there? What it will code for? Quickly, look and tell. AUU. AUU
It's
codes for
ILE.
ILE. It's called isoleucine. Okay. ILE is the short form. Okay. There's a 3 alphabet short form, 3 letters short form. So you're right. AUU codes for isoleucine. Now let's say there was a mutation that happened and it became AUC. What happens?
It remains same.
Exactly. There is a protection there because the code is degenerate. That's called the degeneracy of code. Okay. Even again, if it becomes AUA, it will still remain isoleucine. But if it becomes AUG, that's a problem. It will become methionine. Okay. One very interesting thing is, look, can you tell me what all codons code for MET, methionine? In the whole table, whoever will find me all the codons for MET, tell me quickly first. What all codons code for methionine?
AUG.
Just one. There is only one codon that codes for methionine, that is AUG. Okay, and that's called a start codon. Make in your textbooks, just highlight it. AUG is the single. So methionine, which is an amino acid, it's only coded by one codon, and that is called start codon, which means this codon is present at the beginning of every protein. That's why it's called start codon. Translation starts here. Is it clear, everyone?
Yes.
So the first amino acid in every protein, every functional protein, is methionine. Okay. So the ribosome, when it attaches to a mRNA, it keeps sliding the 3' from the 3' end unless it encounters the first AUG, and that's where it starts translating. If you mutate AUG into anything, the translation will never start from that site. Okay, that's very important. That's called start codon. And you will see there are 3 codons which are here: UAA, UAG, and UGA. It's written stop in front of these 3, right? These 3 codons are called stop codons, and they do not code for any amino acid. UAA, UAG, and UGA. Okay. So degeneracy provides you with some protection against mutations that very easily happen at the 3rd position. Sometimes it prevents the protein from changing too much, but sometimes these changes can be very lethal. Now imagine if a UAC, which is coding for tyrosine just above the stop codon, the C gets converted into a G, the protein will stop right there, right? And if it is in the middle of the protein, the ribosome will only form half protein, and half protein is not functional, or even worse, we don't know if it might have another problematic or dangerous function, or it acts as a problem in this. So misfolded proteins or half proteins or misformed proteins are also very, very problematic. They cause a lot of disorders and diseases in body. Okay, so this arrangement where 64 codons are there, out of which only 61 codons code for amino acids, 3 codons do not code for any amino acid and are called stop codons. This is called the codon table. Okay, and these are the salient features which we will quickly go. There are 6 features. The codon is a triplet. 61 codons code for amino acids and 3 codons do not code for any amino acids, hence they function as stop codons. Some amino acids are coded by more than one codon, right? Hence the code is degenerate. Degenerate means even if you have a different codon, you can put the same amino acid there and make the same protein. That's the benefit. So the code is degenerate. The code is read in mRNA in a contiguous fashion. There are no punctuations. Yes. So when the ribosome reads an mRNA, there is no, after AUG, there is no comma or anything to tell the ribosome. So it will read AUG as one codon. But if because of any, we talked about mutation in the last chapter, right? If because of mutation, something gets inserted here or this G gets removed, now the ribosome won't be able to read the start codon at all, and it will make a faulty protein. That's why mutations can be lethal as well. So no punctuations. The code is nearly universal. The word nearly here is because we now have figured out that in some organisms, the code differs a little. Some organisms do not have AUG as start codon, but CUG, let's say. Okay. So CUG, otherwise, in other cases, what is CUG? Can you find CUG? Leucine, right? So sometimes instead of methionine, but that's very, very rare. So it's nearly universal. For example, from bacteria to human, UUU will code for phenylalanine. Some exceptions to this rule has been found in mitochondrial codons and in some protozoans. So just remember this. Biology is a science of exceptions always. Fifth rule is AUG has dual function. It codes for methionine and it also acts as the initiator codon, or simply called as start codon. So every protein translation starts with this. And the three stop codons are UAA, UAG, and UGA. So is the code, genetic code, codon, how they figured it out, everything is clear to you?
Yes, sir.
Yeah. Okay, so go back today and revise this portion. Okay. And without a proper understanding of this, it will be difficult for you to understand translation. But if you understand this, translation is just an easy peasy job. Okay. Okay, cool. So I will be teaching you about operons in the next class, translation and operons. Okay. And the last part of this chapter is the Human Genome Project and DNA fingerprinting that we will then finish in other class, and this chapter will be over. So 2 more classes and this chapter is over. So please bring your doubts if you have any, and I'll see you all in the next class then. We'll start the new topic in the next class. Is that okay, everyone? Any doubts, any questions? Alia, Aisha, Iram, and Mariam? No? Okay. See you. Bye-bye.
Thank you, sir.
Thank you.